Hello everyone and welcome to today's live broadcast, the Research Resource Identifier Initiative, What is Needed for the Scholarship in 21st Century, presented by Dr. Anita Bandrowski, a scientific lead at Neuroscience Information Framework Center for Research in Biological Systems at University of California in San Diego. I'm Susie Valdez of LabRoots and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to invite everyone and remind them that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on that green Q&A button located at the lower left of your presentation window and type your questions into the box that appear on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also note that you're viewing the presentation in the slide window. To enlarge that window, just click on the screen icon located on the lower right. If you have any trouble hearing or seeing the presentation, click on the support button at the top right or use that Q&A button and let us know we're having a problem. That presentation is educational and it offers educational credits. Please click on the button in the lower left window and follow the process for obtaining your credits. Please join me now in welcoming Dr. Anita Bandrowski. I will now turn the presentation over to her. Welcome, Dr. Bandrowski.
Hi, welcome. I hope everyone can hear me okay. Thank you so much for that wonderful introduction. I really appreciate it. So thank you again so much for coming, everyone. And I would love to talk to you today about key biological resources and the RRID system. And this is really one of the things that is really needed for scholarship in the 21st century. I mean, we are going to be moving on. We have these web-enabled technologies, and the goal is to bring scholarship into that 21st century. So the topics that I'll be covering in this presentation is what are these RRIDs. I'll talk a little bit about the past, present, and future of these. The RRID for authors, so how do I make my paper better? How do I make my grant better? And I'll talk a little bit about the new guidelines from the National Institutes of Health. RRIDs for publishers, so again, there are some reasons why a publisher, a journal would want to join the RRID initiative, and I'll point out some of those things. And then, of course, if you write your own tools, if you create a mouse or an antibody, if you create a software tool, how do you track impact? And so that is the kind of last section of what I'll cover. And I will try to show you and convince you that RRIDs are actually a really good way to go for tracking impact and making your research a little bit better, a little bit more reproducible, a little bit more rigorous, and a little bit more transparent. Okay, so RRIDs are backed by two major NIH initiatives, NIH projects. The first one is the Neuroscience Information Framework, or also called MIF. It was funded in 2008 as an NIH Blueprint initiative, and it established a lot of the aggregation platforms that we've been able to use since then to actually enable this a lot of the 21st century scholarship that we are really working and pushing for now. It created a lot of comprehensive data registries, including those for antibodies and also a lot of digital projects such as databases and software tools. And then there's the NIDDK Information Network, also called dkNET. That was funded in 2012, and what it did is it allowed us to create a very generic platform that allowed us to also customize any version of any of the data sets that we had and display them in a specific way. So it expanded a lot of the data indices, it expanded the reach to become much more consistent with a broader biological project as opposed to simply neuroscience. So what we aim to do really is to actually identify and monitor the scientific food chain. And now, when we look at this, the question really becomes, okay, why is there a picture of a chicken up here? Actually, it's a duck. All right. Well, research resources we think of just like ingredients in a recipe. And so here on the left-hand side, what you'll see is that there are some, incredibly lovely recipes. So, whoops, there we go. There's, duck innards, and there's sugar, there's soy sauce, both light and dark, and all of these things. And you can see that in this particular recipe site, not just there are a bunch of images, which I would call the results, but also there's a lot of ingredients, and these things are actually linked. But research resources, things like antibodies, organisms, cell lines, that's an important one, and all kinds of other tools are these sorts of things, but they're very seldom linked. And so what we'd like to do is maybe, essentially bring our ingredients of the scientific variety into the 21st century, as we said. So in the age of global search, we should be able to easily answer just two extraordinarily basic questions, and we should be able to do it across all of biomedicine. These two questions are, what resources were used to produce the results of this particular study? Which antibody, which cell line, right? What are the other studies that have used this particular antibody? We should be able to answer that. But it actually turns out that even with Google Scholar, we really can't answer this question very well or at all. Now, this kind of question is really important for rigor and reproducibility. It is very important for tracking the impact of these research resources. If I made a mouse, I want to know who used it. I want to know how they used it. The same thing for anyone using a software or creating a software tool. We want to give credit to that resource creator. We also want to aggregate data. We want to understand who else used that antibody, because if I'm having trouble with that antibody in my lab, then I want to see all of the papers that use that very specific antibody so that I can figure out why it works, why it doesn't work, right? To monitor the health of the scientific food chain is the last piece. So if there's a problem with a cell line, how do we know that? How do we know when we're reading a paper that there is actually something that's reported about that cell line well after, potentially, the paper was published? But now we know that this particular cell line was, let's say, transformed and became a HeLa cell, and that happened at the manufacturer. It didn't even happen in the lab. And we know that. Maybe we ought to know that when we're reading a particular publication. And why can't we do this now? So it turns out that scientists actually often, and I'll show you this, don't put in enough information into the paper to make the research resources, the antibodies and the software tools and what have you, easily identifiable. And there are lots and lots of different ways in which we've actually chosen how to talk about a particular thing. We are incredibly, incredibly creative when we talk about all kinds of things, including antibodies. But that actually doesn't help us find these things easily. If we all said exactly the same thing, right? If we all said it in exactly the same way, it would actually be really easy to find, but we don't do that. We are very creative people, so we will write these things in a very creative way. In fact, we are so creative sometimes with our nomenclatures that no one can find anything. So let me show you a really easy example. So here's a nomenclature for a mouse that this particular author is using. Now, this particular author calls this mouse the NOD-PK SCID IL-2 receptor gamma chain null mouse, and they tell us that they bought it from Jackson Laboratories. So as a, reasonably aware of the web kind of a person, I go and I type that into the Jackson Laboratory website and I get back nothing. Maybe with antibodies it would be better. So the same author and many, 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 many of his friends actually discusses that they this author used FITC anti-human CD19 from BD. And they tell us it's in Franklin Lakes, New Jersey. And so as a human being, I go to the BD catalog and I find no products that actually match this search. Right, so that's a problem because these two things that are the basic ingredients of this particular study are not there. Now, this is not the only author that actually does this. In fact, when Vasilevsky et al. In 2013 looked very carefully across all of biomedicine, all of the different subfields of biomedicine, a lot of the different impact factors of journals, and she went ahead and she looked at this entire set of data that was just basically her going through and trying to find these antibodies, find these cell lines, constructs, and organisms. And she said, could I find this thing? And in 50% of the cases, she could not find the antibody or the cell line or other things. So we are, as a field, really bad at doing this. But can we fix this? Of course we can fix it, right? And the question is, how do you do that? Well, instead of putting, whatever lakes, right? How about we just put in an identifier? And then that identifier will actually tell us what is that thing. So one identifier pictured here is AF1494, which is catalog number. Of course, that's not globally unique, but I'll show you that in a second. And then another one is this RRID, and we will talk a lot more about this. So the history of these RRIDs, what are these things? Well, in 2009, we thought, hey, there's a problem. We can't find enough information in papers. And then moving on down the line, we had many starts, and some of them worked out. Some of them really didn't work out. We thought we learned some things back in 2010 and 11. We actually reported our findings about these catalog numbers to the entire editorial board of the Journal for Neuroscience. They use a lot of antibodies, and they said, wow, that's terrible. And a year later, nothing had changed. So we said, okay, well, it's not enough just to say, hey, this is a bad thing, right? We should do better. No, actually what we need to do is we need to create a system which allows the journals to come in and basically make this better. So how do we do that, right? So what we did is we actually followed up. We created a whole system in order to make this really easy for the journals. We had their input, we had their buy-in, we had them all in a workshop at NIH, and it was great because they got to say, my problem is that I have all of these different things that I need to worry about. So how can you help me solve this problem to make methods better, to make them more transparent, without the author having to go to 50 different websites? Right? So then we put together this portal which aggregates all 50 things that they needed to get into one place along a uniform search, and we started in earnest in 2014. We launched the RRID project, and we were able to essentially move forward from there. So from that point on, we were adding journals, and I will show you some of that. Okay, so just at a very base level, what are RRIDs? So the RRID is sort of like a unique barcode, and that unique barcode is for things like mice. So here is an MMRSC mouse strain data sheet, and you can see that there's a citation identifier with the full RRID syntax right there. Also for things like antibodies. So again, this is a product catalog from BioLegend. And there is the RRID for this particular antibody. So a scientist who wants to do an experiment, there's a little pointer thing. Where is that? Okay, so if you look up here on the upper right-hand side, you see the scientist who's performing an experiment, and that scientist will actually go in. They will look for these particular kinds of research resources. So they'll they'll buy their mouse, they'll buy their antibody. They will do the experiments that they are going to perform to ask a scientific question about those, which involve these research resources. And then when they go to report, the publishers, including some that you can see here, the Society for Neuroscience, Endocrine Society, eLife, Cell Press, and others, will actually ask these authors for those identifiers. And then when their paper comes out, you can start to do interesting things. So remember that recipe? Well, here's a paper. And this particular paper actually links back to this antibody. And that is additional information that somebody reading this can click on to then go back and find more information about that particular product. So others, so Elsevier has actually done even more. They've actually created an app that displays the antibodies that are used in a particular paper. And though that app actually contains more information, so we know what the vendor is, we know clonality, we know some of the other things about this antibody, and that just pops up on the side of the paper. So we have many ways of naming these things, right? We have the name itself, whatever that name is for that reagent or resource. We have a catalog number, a stock number, and then we have this RRID. Why do we need this thing, right? Why do we need an RRID when we have stock numbers? Well, the stock number is 025, right? How do you know that's a volume versus a mouse? You may not. The names of these things are actually extraordinarily good. They're very precise, but it's actually very hard to follow for many authors how to actually properly assign or understand the nomenclature. And the RRID is a little bit, maybe somewhere in between a stock number and a name. It definitely has a syntax. The stock number here is locally unique. So if you ask a particular catalog, hey, do you have the 100L? That catalog will be able to tell you, hey, I do, and here it is. If you don't know which catalog to go after, then that 108L or the 225 or the ZL1515 is a little bit more meaningless. So there's something called locally unique, and then the RRID is actually based on a lot of these, not always, but it's actually globally unique. So you can actually ask a very specific question of Google and you will get back answers about these. So the stock number is essentially for customers. The RRID is for authors and also for search engines. So this actually helps both of those kinds of characters. And the names are really for curators or for researchers that are very well versed in nomenclature and have been trained on understanding nomenclature, which is unfortunately not as many people as we need. So the name actually will tell you what is this thing, what are the genetic differences between this particular animal, and this is a mouse, and another mouse, right? So this will actually tell you things about the genetics of that mouse, whereas the stock number will tell you where you can buy it, and the RRID is how do I cite this thing so that it's recognizable by machines and also all of your friends. And of course, they all have to work together. So if we look at the actual anatomy of an RRID, we have this RRID portion, which is I use this entity, I use this thing in my study. It's not I'm talking about it, I'm using it. Then the ZERC here is the repository or the authority. So ZERC is where you go to buy this other local identifier. So there is a ZL1515, and this happens to be a fish. But the entire syntax is demonstrated here. So if we look at the entire syntax, we're actually looking at Millipore, which is the company or the provider. We have the local identifier here. We suggest that the author always uses the lot. Some do, some don't. But then there's the global identifier, and this comes from a repository and a registry for this kind of reagents. So in the case of antibodies, like is listed here, there's the antibody registry, and that is the antibody registry identifier. So what should authors do with these things? Well, in the author's workflow, what the journal does is they direct authors to this particular website. So this is a SciCrunch portal. And they are going to search for their particular research resources. So in this case, 108L is the thing that I'm searching for. And what you can see is that this is the eyeless wild-type salamander larvae, Ambystoma mexicana. And here's a little bit more information. You can click on that button and you can actually get back to the Ambystoma Stock Center. And this is how you cite that particular salamander. So if you click on that, copy, paste this into your paper, then the paper is published. And now once the paper is published, what happens is it actually becomes data. So then somebody else, who's not you, can go in and they can search for, in this case, this is a mouse. They can search for the 664 mouse. Excuse me. And this mouse happens to have been already published in 147 different papers. Sorry. So, is this a hard thing to do? It turns out that when asked, the authors at eLife were asked to fill out kind of a brief survey just to figure out, okay, now eLife is making everybody do this, and, you know, is it really the bad guy for making them do this? But it turned out that all of our responses were really positive from authors, and they said really nice things like, you know, adding the identifiers actually turned out to be very easy. So that was from one paper, with the authors of one paper. And another paper basically said, well, you know, besides helping other labs to actually reproduce our own work, we don't always know what all of the different reagents were, so this is a really good way of just making sure that we keep track of exactly what's used in this paper so that it's there for lab members in the future. So again, these are really nice things that basically can you know, can help not just other labs but also potentially our own labs in the future. So the other reason why this is a really important thing to do is if I am an author, I am about to publish my paper. Maybe I checked with the ICLAC authority before I bought my cell line, and maybe I didn't. But before I publish a new paper with a particular cell line, I may want to know whether the authoritative source on contaminated cell lines has said anything about the cell line that I'm using. And let me make sure that this is clear. There are cell lines that you can buy today that are actually contaminated. There are over 700 of them. 700 cell lines are not actually what they say they are before they get into your lab. Now, we always know that there are also antibodies that have problems and other things, but this is a really clear case where, at least before you publish the paper, that information should be consulted. And the nice thing is, when you go and find your RRID, you have that information right there. So here I have executed a search in the resource identification portal for cell lines, and this is the 15C4 cell line. And I didn't say anything about this being contaminated, right? But what comes up is, hey, this is the cell line, here is the repository, and look at that comment, problematic cell line, it's contaminated, this is shown to be a HeLa derivative. And there actually, if you click on this and you go out to Cellosaurus, that aggregates a lot of this information. They actually work with this ICLAC community to make sure that that information is in as soon as it comes out from ICLAC. The information is right there. So when somebody is citing this particular cell line, they will know. that at some point in the past, the ICLAC community had found that that particular cell line was contaminated. So again, that could save you a retraction. Now, why should journals care about these RRIDs? So looking at it from the perspective of a journal, one of the things that it does is it actually takes all of these different critters. So we saw ambystoma. I think we saw a couple of examples of mice. We saw some examples of antibodies and even cell lines. But everything is all in one, including software tools here. The fish, the rats, we even have pigs and pond scum. But all of these things are sitting inside of one place, one portal. So the authors that you're asking to do this only have to go to one place, and the format is very consistent. So always there are notes, there are comments, there is a cite this button, or again, there's a cite this field in all of these things. The button is kind of covered over by how many things we have, but it's there in all of the in the portal, so that it's very consistent across every kind of organism, across every kind of research resource. We work with, again, we work with all of the model organism communities, the big ones, and we're more than happy to work with others. But there's not just one database, there are actually two. There's a master copy, so that when a particular fish is no longer sold, it's fine, right? The antibody manufacturers don't always sell the same products. But if you're going to buy that product and then use it for scholarship, there's a different thing that you need other than the catalog. So the thing that you need is, hey, this thing was there, I used it, it was used by these 12 other papers, and now it's gone. So in this case, this particular fish is no longer indexed by source database. This is gone. You cannot buy this right now. But that's okay. You may have it in your lab. You may have used it in your experiment. You should be able to cite it. It shouldn't matter that a particular vendor doesn't have that thing anymore, right? Because we still have the data. So that's the comparison to the master copy. And that information continues to be there. It continues to be resolvable. It's kind of like the library card catalog. You know, you don't have the book, but at least you know it was there. Okay, so again, RRIDs are they are persistent. So this is what a persistent unique identifier does. So it lives there. It's the card catalog-like thing, which tells you there was a fish. This is what it was called. This is what we know about it. Here are the papers that were published with it. But maybe you can't get that anymore. Okay. So the other thing that it does, when we went back a couple of years later, and we really looked deeply at the first 100 papers that were published with RRIDs and asked the question that Vasilevsky et al. Asked 2 years before. And we said, did this make a difference in that journal, in those papers? Did it make a difference to ask for RRIDs? And in fact, this plot right here shows you that, in fact, it does make a difference. It makes a huge difference, right? So antibodies were identifiable. These are now looking at the same journals, just the issue before RRIDs came out. And then the same journal with the issue just after people were asking for RRIDs. And what you can actually see is that the identifiability, so can I find this thing, goes way up. That goes way up. So what we are really doing, and this is a great paper with these nice links to these particular mice, what we're really doing is we're getting rid of the citation format for this mouse, which looks like this, you know, ERC mouse from Jackson Laboratories in Bar Harbor, blah, 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 right? And we're shortening it. So this is a better because it's shorter format. And it also will give you the actual mouse. So the added benefit of you can actually find the mouse, which is very nice. Okay. Now, what if you made a mouse? Or you made an antibody? Well, it turns out that if you make a mouse or if you make an antibody, and someone did make this antibody, what you can do, if people cite it very specifically, then you can actually pull back potentially all the papers. If we can you know, get this instituted in enough of the literature, we can pull back the bulk of the papers or all of the papers that are actually using that antibody by talking about it very specifically. Sorry. And there is no need, because this is a Google Scholar, I'm not even logged in, page. I could just look at this, put this ID in, and get back all of the data. No need for fancy tools, no need for natural language processing. But when people use identifiers, it actually does more things, right? It's not just I can put it into Google Scholar as a search string to make it unique, but it actually, what it does is it starts to become an ecosystem of identifiers. And that ecosystem of identifiers, you know, there are a lot of them. The papers, each paper has an identifier, actually has several, right? So potentially it's got at least a DOI and a PubMed ID if you're going to be indexed by PubMed. Probably has a PubMed Central ID if you're going to put it into PubMed Central. And these identifiers make the paper unique. So if there's a version on my desktop, there's a version on an HTML web page, and there's a version that's a PDF, and there's a version over in PubMed Central, these identifiers actually tell me, hey, that's the same paper. It's kind of important. If you look at something like an author, it turns out I actually published a paper with somebody whose name was y.li, L-I. And all of the websites, for some reason, could never, ever get to the right person. There's a lot of Lees. Not as many Bandrowskis, but there are some. So a lot of our names are not unique, right? So we have now Orchids, and Orchids are very, very useful because they differentiate that y.li or that a.gupta or, you know, the Martone or the John Smith or whatever else, right? So we want to be able to differentiate the person that we're actually talking about. That's the author. It's not, you know, some generic John Smith, right? It's this one. It's this one specifically. And so again, we have for authors, we have Orchids. For papers, we have DOIs. Funding, so grants absolutely have numbers. Data sets and code often have DOIs. Books have ISBNs, but research resources actually have RRIDs, and they work very similarly to these papers. They work very similarly to the author IDs, so the Orchids. So the goal of the findable, accessible, interoperable data, right, and findable, accessible, interoperable research resources is to link different kinds of artifacts together, including people. What we want to do is we want to have all of the evidence where a claim is based on a whole set of procedures to allow us, right, to now link back to all of these sections which we do know and we know very, very well. Okay, and we want to do that by using unique identifiers. That way we can actually start to create interesting graphs. Who was that person? So not only for disambiguating, what was the funding, which papers were produced, and which resources were used. All of these things need to actually be utilized in order to trace the utilization and trace the impact of what we're doing. So again, if I make a mouse, and nobody tells anyone else which mouse they're using, no one can find it anywhere, I can't report back to my grant, to my funding officer, hey, somebody used this mouse, but I have no idea who. Maybe they'll write me a nice letter. But that's not repeatable, that's not traceable. So we want to use these identifiers so that our impact is actually traceable. And let me just show you something kind of fun. So when we have this kind of structured data, or no, not even completely structured data, but we have these articles like this with these identifiers. This is a mouse. What we can do is actually we can read those by using robots. And this is my little picture of a very cool reading robot. And so if you look at this, if you click on it, you can actually get to a page that looks kind of similar to this. But what I can do with my little 15 lines of Python robot is actually I can put the same information back into the paper. So now I'm reading along, and this is a particular knockout mouse, and this is what the database says about that mouse. And if I click on that, I actually get back to the stock center itself. So I can actually know much more about this particular reagent than somebody who's just reading the paper. And so this robot is actually pretty well described here in a blog from John Udell. And there are a bunch of examples. This is one. So PubMed Central, if you just type into PubMed Central, 410 7038, you'll get back to a paper that has an example of what one of these things looks like. So you can certainly do that, and I urge you to explore these kind of robots and these kind of overlays into the literature. So what we do with a lot of this robot reading is actually we take the papers, we process them by having the robot actually help our curators to read the paper, brings back the information, and now we've made an assertion, or we've exposed an assertion about a paper which has an identifier and a tool that has an identifier. So in this case, this is the Abyss tool. And this Abyss tool now is associated with a particular paper. And this paper is, again, from GigaScience. So GigaScience has published a paper. This is the title of the paper. It's by Fu, Y. Fu. And then this paper is now associated with the record for that particular software tool. So this information then flows, and it flows very quickly to the tools. And so now if I'm the tool maker for this particular tool, what I can do is I can download all of my citations. So this is a citation index that comes straight out from just the person essentially citing this particular tool using the ID. And again, that can happen normally by somebody reading and saying, oh, wait, that is the Abyss. But because this can be read by robots, because this can be read very, very quickly and automatically, then what ends up being able to happen is that that information can be automatically pushed. So if we look at how many RRIDs there are over 4,000 papers now. We're very soon to hit 4,500, which is not a huge amount yet. The number of journals that we have is currently at 349 different journals. And the types of tools, so if you look at the donut ring here, most of the things that are being cited are actually antibodies, and that's just by number. Tool, it just refers to software tools or databases, and cell lines, which came on a little bit later to the project, are here in green. And then, of course, lots of mice, some flies, and then a smattering of other critters. But if you look at the number of papers, the number of times that one of these things is used, the most useful tool, apparently, according to our subset of 4,300 papers, is the ImageJ tool. The next one is GraphPad Prism, MATLAB, and there's R and SPSS. And then down here you actually see that mouse, which is the C57 Black 6 mouse. Actually turns out to be very, very popular. Everybody likes that mouse. If you look at the antibodies, actually this is the most popular one. That's one from Millipore, and there's a couple from Thermo down here. Most of these antibodies that are used in many, many, many papers are actually secondary antibodies, but this list continues for about 50,000 rows, so it's actually quite long. But so if we look at one of these tools, now we can look at one of these tool pages. So this is ImageJ. So if I had created ImageJ, I could do various things. I could actually log in to edit this particular page. So if I search in the RRID portal, I type in ImageJ, I hit enter, and I get into ImageJ. Here there's a lovely description, and I say, oh, no, that description is all wrong. Well, what I can do is I can change it. Right? There's also a bunch of information in these tabs. So there's kind of the basic information. These are keywords, your grants, and all of that. There's a bunch of relationships. with other tools. So if you were part of a university, there would be a relationship with the university. The paper about the tool is here. So, there's a specific idea. I made this tool, and here I write a paper about it. Great. That doesn't always happen at the same time. In fact, often there's a pretty big disconnect. So for this particular tool, ImageJ, actually we had some interesting analysis of this, because it took many, many years, like over a decade, for somebody to write a paper about this really useful tool. And by that time, it had already appeared in hundreds of papers. But then there's this section here, which is the referenced by. So if you click on that, you will see how many people are talking about your tool. If you have your tool, you can see that particular component. So again, you can also claim ownership of this tool. So I made this tool, and there can be multiple owners, just like for every paper there can be multiple authors. And then you can actually subscribe to new mentions. So if there is a new paper that comes out and it says, hey, I used ImageJ, then next time you log in, you would say, hey, looks like this paper actually talked about your tool. And that's kind of cool. The other thing you can do is you can go into your profile if you've actually created one, and you can, just look at the name. So this is my name, and here's my Orchid. And that Orchid, so there's a button here that says associate your Orchid, and if you have an Orchid, great, good job. I congratulate you, and great. If you don't have an Orchid, I really recommend getting one. It takes maybe 2 minutes. But what it does is then it starts to aggregate your funding information, the fact that you're at a particular university, what you've published, all gets fed into your Orchid profile. And this Orchid profile can be used by institutional review boards. Maybe it will, maybe it won't, but it's one of the options that tenure and review committees can actually get information from your Orchid profile. So Orchid is great for that. And one of the things that we're really working on right now is to connect up, again, the fact that I might own a tool. I may have created a tool, so let's pretend, I didn't do it, but let's pretend that I made ImageJ, and what I would be able to do is I would be able to claim and actually move to Orchid the information that I made a particular tool. So in this case, this is my Orchid, and here's my Orchid page. And so my Orchid page, there's Anita Bandrowski, that's nice. It all kind of more or less fits in, AE versus Anita. Well, could that be me? Yes, it in fact could, because there's my ID. So there is the ID, and then what you see here is that there's the antibody registry database. Now this is not a paper. In fact, we have not yet written a paper about the antibody registry database, but we have created the antibody registry database, and it is in use. Trust me. Just see a couple of slides ago. So, this database is being used, but it doesn't yet have a paper associated with it, which is actually fairly common, apparently, in the database world. So, what I have for it, though, is an RRID, and here it is. And so now this sits inside of my Orchid profile. Now, if my tenure committee wants to use this information, they can. And in fact, we're working on an integration with Orchid so that we can do this and ask authors of tools whether they want that information to be pushed to Orchid. So if you've created a tool, a software tool at this point is how what we've enabled. You can do that today, and within some months we should be able to enable and allow for both antibody creators and then other research resource creators to kind of do the same thing. So we'll need to get this all worked out, but this is where we want to get to. We want to say there are parts of intellectual work that are not papers, and those should be accounted for in some way. Who chooses how to do that? How to evaluate a particular scientist and their intellectual output based on that particular type of work is up to them. But for us, we want to push this information out there. we want to give them the ability to judge people not just on the papers they've written, not just on the abstracts that they've presented and put in for conferences, but given the fact that, hey, maybe somebody made an antibody that then allowed cancer to be cured. Well, great, I think that person deserves some credit for that. Maybe not a curing of cancer, but some credit for that should be given. So the other things that we're working on in terms of putting this data out there about these papers. So for example, this again is a project from the CrossRef folks. And the CrossRef folks now have data about events that happen to a particular paper. And those events can be many, many things. They can be somebody tweeted about the paper, somebody wrote a blog about this paper, somebody downloaded this paper, and all of that. And so one of the events is, well, we had a reading of this paper by this automated tool when a curator opened the paper. Then that was associated with a whole bunch of things, including, oh look, there is a mouse. Right, so this mouse is part of now the record for this particular paper in the event database. So we work currently with Hypothesis, which has a W3C compliant annotation standard that allows all of this all of this kind of data to flow back into CrossRef. So now CrossRef, if you guys are not familiar with CrossRef, CrossRef is the kind of provider for most of the DOIs. So if you click on the DOI, that little link that takes you to the paper, that's provided a lot of times by CrossRef. Okay, so if we're going to look at sort of how many journals and how many papers over what period of time that we're actually seeing, what we see is that since we started the project in 2014, where we started with 25 journals, we currently have over 300. We started with, or actually ended the 2014 with 115 papers. The rate of increase is roughly doubling. So if that continues, which it appears to be so far continuing, it will take us between 5 and 10 years to kind of be a much more pervasive set of identifiers in the literature. We will, by I think 7 years at this rate, be in all of the 23,000 journals that are in PubMed. So we look forward to that. These are currently the top publishers. So if you look at Wiley is on top, and Cell Press is the third one. Ooh, I'm not sure what happened. Okay, I think. There we go. Okay, so I just wanted to kind of wrap up and say there are a lot of papers being published every single year in PubMed, and the reason that we've really started to invest a lot of our energy into building automated systems, building some of these robots, is because really when you want to improve tracking, when we want to improve how we move data about papers and how we allow different kinds of data to go from one place to another about individual papers. So for example, who cited whom is something that we kind of understand, but who cited which mouse is not yet as well developed. So what we want to be able to do is we want to inject machines as much as possible into this equation so that the tracking and the impact of resources becomes a more simple and straightforward thing to be able to do. We want to help authors actually do this better. We want to provide more of this credit to tool makers, and we want to improve the way that they do this. reduce the barrier to entry. We want to be able to also inject some of that information into papers where it's, where there's clearly something where the reader might want to know, hey, when they publish this paper, this particular cell line may have been okay, but at the moment we understand that there was a contamination event that was reported, so we're not really sure. So, take this with a grain of salt, which everyone should anyway, but having that information right there in front of the reader tends to make this process much easier. So we want to provide this information to authors. We want to provide it to readers as well. Okay, so is there anything that you can do? Turns out you can actually add your tool. If you have created a tool, you can use the RRID in your methods section. Please do. If your journal editor asks you for doing it, please do it. If your journal editor does not ask you, it turns out most of the time you can just get them in there anyway. So those things just help us track, and it's a fairly straightforward system. When you review a paper, whenever I review a paper, I look at the methods, and I look at the methods because I have to look for the RRIDs. And it turns out when I talk to my editor colleagues, when they review for RRIDs, they look at the methods more frequently, and they have actually told me that they've caught really big problems because they were looking more carefully at the methods. So this is just one way. Again, it's a shortcut to look at the methods and to really review the methods instead of just reviewing, what the author actually said. And of course, if you have a journal, please talk to me. My email is at the bottom, Bandrowski at UCSD. Please talk to me about it, and I would love to. I'd love to be able to come together and say, hey, we can do something for your journal. Okay, so thank you, and I really appreciate your attention. I believe that, excuse me, I believe that we're going to now cut this off because I believe that I have run out of time. Okay, thank you.
Thank you so much, Dr. Bandrowski, for that informative presentation and your important research. And thank you to the audience for participation and questions. Dr. Bandrowski will be emailing answers to those participants who sent in questions. I'd also like to take the opportunity to thank LabRoots for making today's educational webcast possible. Before we go, I want everyone to know that today's webcast will be available for on-demand viewing through December 2017. You'll receive an email from LabRoots letting you know when the webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thanks for joining us. We hope to see you again soon. Bye-bye.